condition that can cause red blood cell to become a sphere. And other than that, we have hereditary elliptocytosis. So here the shape is different. It is not like a sphere. Now the cell becomes a elliptocyte with a oval shape. So those two defects are hereditary defects, inherited defects, uh, causing hemolytic anemia. So when the shape of the cell changes from the biconcave normal shape, the macrophages in the reticuloendothelial system, they identify uh, such cells as abnormal and undergo destruction, causing hemolytic anemia. Then we have metabolic defects as well, inherited metabolic defects, G6PD deficiency. You might have heard under biochemistry. Uh, this is an enzyme in pentose phosphate pathway, and that is the only pathway reducing energy inside the red blood cell, because red cell doesn't have nucleus and mitochondria. So, if there's a deficiency in this enzyme, T6PD, glucose 6-phosphate dehydrogenase, the producing energy will not be produced. So, will be produced at a lower amount. So, it depends on the severity of the deficiency, and it can result in lack of generation of reducing energy, and lack of ability to fight with free radicals producing inside the red blood cells. So during the metabolism of red blood cells, many free radicals can be formed, and they can cause oxidative damage to the red blood cell physically. And these reducing energy, and especially the antioxidants need reducing energy. So if you don't have reducing energy, the antioxidant, they are unable to reduce the free radicals, and they are unable to prevent the oxidative damage, causing structural changes in the red blood cell. And then again, if you have structural changes in the red blood cell, the macrophages identify them as abnormal cells, target them, and destroy them. Similarly, a pyruvate kinase, an enzyme in glycolysis. So, when there's a lack of pyruvate kinase, there's a reduction in the production of ATP. ATP is the energy source it requires for the proper function of sodium potassium pump. So when you have a low amount of ATP or no ATP at all, the sodium potassium pump is unable to operate, causing the leakage of potassium out of the red blood cell. Uh, so it results in accumulation of sodium and dehydration of red blood cells, again causing structural change. So these structural change is the triggering factor for macrophages in the RE system to identify such cells as abnormal and destroy them. So then we have hemoglobinopathies and thalassemias. Hemoglobinopathies are structural changes in the hemoglobin because of inherited abnormality. Examples include sickle cell disease, hemoglobin C disease, hemoglobin E disease, D disease. Those are hemoglobinopathies. And then you have thalassemias. So in thalassemias, you have reduced production of one type of uh, globin change in hemoglobin. So hemoglobin, adult hemoglobin, is composed of two alpha chains and two beta chains. So in thalassemias, because of inherited mutation, genetic defect, there could be either lack of production of alpha globin chains and or beta globin chains, causing beta thalassemia and alpha thalassemia. So based on the defect, deficiency, you can categorize them as thalassemia major and thalassemia minor, and also in between level called thalassemia intermediate. So we have beta thalassemia major, beta thalassemia minor or trait, beta thalassemia intermediate. Similarly, alpha trait, alpha thalassemia major or hemoglobin hydroxylates. So, okay, so right, that's the hereditary classification, and then acquired classification. So, acquired hemolytic anemias, you can divide into autoimmune and alloimmune. So autoimmune hemolytic anemias are now these acquired hemolytic anemias are rare in comparison to hereditary hemolytic anemia. So among the acquired hemolytic anemias, the autoimmune hemolytic anemias are kind of rare. And alloimmune anemias, hemolytic anemias are also rare. So autoimmune hemolytic anemia could be either warm antibody type or cold antibody type. You by now you know what the autoimmunity is because you learn the immunology in the first year. So alloimmunity is the antibodies formed from somebody else's body attacking inside our body. So alloimmunity uh, can cause hemolytic anemia as well. Examples include hemolytic transfusion reactions during incompatible blood transfusion. You can learn more about this on the transfusion hematology, hemolytic transfusion reactions, the different types of reactions, and how do you detect such reactions in the laboratory. And the hemolytic disease of the newborn or fetus can cause hemolytic anemia, alloimmune hemolytic anemia in the developing fetus or the newborn baby. Again, because of the incompatibility of blood between the mother and the baby, usually related to Rh blood group system. And allograft are the transplant graft, including bone marrow transplants. And sometimes the anemia could be drug associated. Certain drugs can cause hemolytic hemolysis in your system if you're under such drugs. Certain antibiotics, including dapsone, can cause hemolytic anemia. So those are acquired conditions. So okay, now you know what are the inherited and acquired conditions. And then we have non-immune hemolytic anemias as well. So these hemolytic anemias also fall under acquired reasons, and under non-immune classification and non-immune category actually we have red cell fragmentation syndrome and infections. So what is the meaning of red cell fragmentation syndrome? Here, red cells undergo physical fragmentation. Fragmentation means red cell uh, breaks into several pieces physically because of a physical obstruction inside your blood vessels. So such destructions are known as microangiopathy, microangiopathic hemolytic anemia. destruction of red blood cells inside blood vessels because of physical barriers, so physical obstructions. Examples include DIC, disseminated intravascular coagulation. It's a condition characterized by formation of many small blood clots inside the circulation, especially in the microcirculation, in your venules, arterioles, venules, and capillaries. There will be formation of numerous blood clots because of a pathological condition. One example is septicemia. If you have profound bacterial infection because of bacterial toxins, the coagulation cascade would initiate and form small blood clots everywhere in the circulation, especially in the microcirculation. So when red blood cells try to pass through these blood clots, small blood clots, there will be physical restriction and obstruction and damage to red blood cells, causing red blood cells to become fragments. You can identify them, these fragments, in a thin blood smear. When you prepare a thin blood smear and stain with Leishman, you can identify them. And such cells are known as histiocytes. Fragmented red blood cells are known as histiocytes. Thrombotic thrombocytopenic purpura is also a similar condition. So here also there will be formation of platelet flux, numerous platelet flux because of the activation of a factor called the von Willebrand factor. We can learn about more under hematology 2 when you learn platelets and coagulation. So I will just brief it here. So same effect in thrombotic thrombocytopenic purpura. There's a massive obstruction in the microcirculation for red blood cell to pass through, causing physical shear damage, and fragmentation. Hemolytic uremic syndrome, again, very similar. The action is similar, final action. It is a secondary condition caused by bacterial infection, especially E. coli. Certain strains of E. coli produce a toxin called Shiga toxin, and this toxin can induce endothelial damage of blood vessels. So this profound damage induce coagulation cascade, so that there will be clots, blood clots formed at the sites of endothelial damage, which is widespread in everywhere of the circulation, again causing the same type of damage. So we have a common name, microangiopathy, for such damage. The second one is March hemoglobinuria. So that is different, but terminology is not fully understandable. When you walk or when you run, when you march, because of this physical action, red blood cells undergo hemolysis intravascularly in your lower limbs. So that is known as, that's why it is known as March hemoglobin. Hemoglobin, the hemolysis, red cell destruction occurs inside inside vessels when you march, so when you walk or when you run or when you do strenuous physical activity. So this is related to strenuous physical activity, right? Okay, and then infections too can cause red blood cell destruction directly. Malaria, dysphagia, cause direct red cell destruction. Thirdly, physical agents to directly damage red blood cells, chemicals, chemical poisoning can cause red cell destruction and burns, severe burns. The heat, if you expose to extreme heat, it can cause red cell destruction intravascularly, causing hemolysis. And hemolytic anemia sometimes could be secondary to other conditions as well, such as severe liver and renal disease, chronic kidney disease, and stage renal disease, and also chronic liver cell disease. Severe forms of such conditions can cause red cell destruction. You know, the liver liver produces proteins required for red cell membrane. The liver is severely affected. There is a reduced synthesis of such proteins, membrane proteins, causing a fragile red cell membrane, which is susceptible to lysis. Paroxysmal nocturnal hemoglobinuria, another acquired disorder causing intravascular hemolysis. So when you have this condition, the red cells are abnormally susceptible for complement-mediated hemolysis. We can learn more about this later in a different sub lecture. Right. What are causes of intravascular hemolysis? We discussed some of these causes earlier. ABO mismatch blood transfusion causing transfusion reactions, hemolytic transfusion reactions, and G6PD deficiency with oxidant stress. Mainly, the hemolysis in G6PD deficiency occurs inside the spleen, extravascularly, but rarely. Occasionally, PD deficiency can cause intravascular hemolysis as well. Red cell fragmentation syndrome, you learn what that is, and autoimmune hemolytic anemias, drugs such as dapsone. Dapsone is given in leprosy caused by Mycobacterium leprae, malaria, paroxysmal nocturnal hemoglobinuria, March hemoglobinuria, and unstable hemoglobin. Unstable hemoglobins are globin chain defects. These hemoglobins, they are really unstable, causing they undergo oxidation. They undergo oxidation and they denature and deposit on the red blood cell. So when oxidative damage damages the red cell, again macrophage identify them and break them, causing destruction. Okay, right. So what are the laboratory findings? So we can see features of what are the analytes that you need to assess. Bilirubin level, the serum bilirubin level, it should be high, both unconjugated and bound to albumin fracture. You have two types of bilirubin, unconjugated and conjugated fraction. So unconjugated fraction could increase when there is a massive hemolysis. So urine urobilinogen is also increased. So that is the reason for urine to have a dark color. Usually the color is pale yellow if you drink adequately. If the color is dark, like dark brown, not dark yellow, the dark brown, it may indicate increased presence of urinogen. However, it must be checked and clearly identify the reason. Sometimes these color changes of the urine occurs because of the drugs that you are on, or because of other reasons such as the food you consume. And thirdly, haptoglobin, serum haptoglobin level. We have to measure the serum haptoglobin level also, and indicates the haptoglobin saturation, and no free haptoglobin in the plasma or the serum. So it's a really good indicator of intravascular hemolysis. And fecal stropobilinogen level also increase. The color of stools becomes dark brown or dark red. Red cell production. So all of these laboratory features we discussed earlier, they are features of increased red cell breakdown. Now, what are the laboratory features of laboratory indications of increased red cell production? The reticulocytosis, you can measure the reticulocyte count. It is high. So it clearly indicates the hyperactivity of the bone marrow and ongoing hemolysis. And bone marrow erythroid hyperplasia is indicative of and also. So you can microscopically identify fragmented red blood cells, and if you see significant amount of fragmented red blood cells in the blood picture, and that is indicative of intravascular breaking down of red blood cells. So we can see schistocytes and sometimes microspherocytes, elliptocytes. So sometimes because of the damage, red cells, the biconcave red cells could become a microsphere or elliptocyte because of the damage induced by usually a macrophage. I said the macrophage can identify red blood cells with oxidative damage, like in the case of B6PD deficiency, unstable hemoglobin, or PK deficiency. Uh, Macrophage identifies such cells and remove part of red blood cells when they travel through the RA system. So then the biconcave cell could become a microsphere or elliptocyte, and you can see them in the blood picture. Or if you see, if the damage occurs directly in the blood vessel because of the shear physical obstruction, you can see fragments. There are other investigations you can do. You can do flow cytometry and identify membrane defects, such as in the case of hereditary spherocytosis and also hereditary elliptocytosis, and also in paroxysmal nocturnal hemoglobinuria. You can identify the membrane defect using flow cytometric studies. So these studies are confirmatory, and they have a high diagnostic accuracy than screening tests based on biochemical assessments. So for further confirmation, uh, we can do genetic testings if required. If other results are inconclusive for confirmation, you can do genetic testing and confirm your thalassemias, other hereditary disorders. Okay, what are the main laboratory features of intravascular hemolysis? So these are the main laboratory features of intravascular hemolysis. Absence of haptoglobin, So that is obvious. Haptoglobin is there to catch all the free hemoglobin if there is a extensive intravascular hemolysis and releasing of hemoglobin, all the haptoglobin would catch these free hemoglobins and become saturated. So you don't have haptoglobin in your serum or plasma. Hemoglobinemia and hemoglobinuria. Hemoglobinemia is the presence of free hemoglobin in the plasma. You can identify that by doing biochemical tests as well as physically using hand spectroscope. Hemoglobinuria is the presence of hemoglobin in urine. It is indicative of intravascular hemolysis. So there is obvious color change in urine. Hemosiderin-urea is a sequel to the presence of hemoglobin in urine. So when hemoglobin comes to the glomerular filtrate, and when when the hemoglobin passes through the renal tubules, tubular cells uh, take, take in these hemoglobins. So they absorb these hemoglobins to the tubular cells, and inside tubular cells, this hemosiderin is a denatured product of hemoglobin, and after a while, about a week or two, these hemosiderin they release, they sloughed off into the uh, uh, filtrate, and there and presence with the hemosiderin. So that in the laboratory by doing iron stain, burn stain, that is